Excellent. So I'm Kristen Bresky. I'm the current acting chair of the Molecular Ecology Working Group, for which our maybe slightly unfortunate acronym is MWOG, which is controversial to be used within the group, whether we call ourselves MWOG or Molecular Ecology Working Group. I'm really excited to be here and get to share with you about what our group does. And I'm joined by an excellent group of panelists that are Molecular Ecology Working Group members: Rachel Gilio, Liz Kirpka, Emily Latch, Rob Lozinger, Cassandra Miller-Butterworth, and Caitlin Otcon. And they're going to be contributing to discussion when we get to the kind of choose your own adventure aspect of this webinar of what topics we're going to dive into in more detail. At that point, I will invite you all to please participate in the Q&A section when we get there. And if there's Molecular Ecology Working Group members online, which I hope there are, please feel free to also join the discussion with expertise and questions and advice. So before we jump into the Choose Your Own Adventure portion of this webinar, which was our title, Choose Your Own Adventure in Molecular Ecology, I just want to give a quick introduction to our working group and the activities that we do and kind of why we were formed and how we kind of serve the broader TWS community. After that, we'll go to the fun part where you're all going to get a poll to take if technology works, hopefully. And we're going to rank these list of topics that were actually selected as topics that we're often asked about. And if you aren't familiar with these terms, don't worry. I'm going to go through each one briefly before you're going to be asked to rank which ones you want to hear about. Then we're going to all come back together and have a discussion about each one of these with a little bit more in-depth information from our panelists. All right. So let's dive in. Molecular Ecology Working Group. Who are we? Well, we were created in 2013. That was our founding meeting at the Pittsburgh National Conference. And we came together really as wildlife ecologists, recognizing that there was a need or sort of gap in the Wildlife Society working groups that could be filled with more of a focus group on questions associated with molecular ecology. And so part of our charter actually addresses these specific objectives of communicating more about molecular techniques and applications in wildlife ecology, conservation, and management, and how these tools can help improve management and wildlife work. Through this, we're hoping to increase awareness of molecular tools in wildlife management, and overall kind of improve broader capabilities and molecular techniques across the entire wildlife society. So this doesn't mean, of course, everyone becomes a wildlife geneticist, but to try to help kind of increase general awareness as well as abilities to talk and think about genetics as they may pertain to wildlife management and conservation questions. And part of this is to be advocates for incorporating sound genetic data into decision making and policy making when it comes to wildlife decisions as they occur. And I think kind of an overarching goal that encompasses what we try to do as a working group is to broadly, enhance awareness of molecular ecology and also awareness of the genetic applications to wildlife biology, and importantly, act as informational and networking resources for TWS and other wildlife biologists. And I think this is really sort of the crux of what we try to do as a group, which is be an informational resource to TWS. So this means, helping bring together agencies and wildlife professionals to apply genetic tools. So how do you form those relationships to incorporate a new technique and a new tool into your tool belt for whether it's wildlife management or conservation? And then also I'm sorry, my phone's ringing. There we go. We also help each other. So trying to stay current with a really rapidly developing field of molecular techniques. And so it's both helping to be an informational and networking resource for the broader society, but then also for molecular ecologists within it. And given this, we have a really interdisciplinary membership of people in a whole host of different wildlife professions, as well as different stages of their careers and education, that come together and kind of help bring the right people together to best answer a particular question with a genetic tool. And so chances are, if you reach out to one of us, if we don't know the answer to something, we know someone who does and could point you in the right direction. So how do we do this? What do we do? So we have symposiums. So here's a list of some of the recent symposiums that we've held. And these range from maybe, direct methodological considerations for the wildlife geneticist, up to broader, more, broadly accessible symposia to help integrate and bring people together to bring genetics into management decisions, which is what our symposium in this November national meeting 2021 is really aimed at. And I'm going to talk about that in a little bit more detail in the next slide, because it's really an exciting symposia that I think is really getting at the heart of what we try to do as MOAG, Molecular Ecology Working Group. We also host workshops. So we've had a workshop that's focused on landscape genetics, and that is a little bit more targeted to someone that might have a background in genetic data and genetic data analyses, and so helping advance the skill set of wildlife geneticists. But then I think one of our kind of staple and potentially most popular workshops that we do every other year and will be at the November meeting. So if you're interested, please sign up. It's Do the Genetics 101, Do the Genetics, and it's a crash course in the basics of wildlife genetics and molecular ecology. How do you store samples? How do you collect a sample? What are the general costs? What are the general laboratory considerations? How do I want to know something about relatedness? What do I do and where do I start? This course is a great way to dive into that. If people that have no experience up to limited experience, or even some advanced experience, to be able to talk with a group of people and work through questions. And so you'll learn how to do the genetics, which is something that we're often asked as wildlife geneticists. We also have student travel awards for traveling to national conferences, students presenting in something related to molecular ecology. And we spread job announcements for jobs pertinent to TWS and WOG members. So I just want to spend just a little bit more time on this year's symposia, the Adapting Management Through Collaboration, Integrating Genetics to Inform Decision Making. And this whole symposia is designed around how do partnerships form between a geneticist and an agency, state or federal, tribal, NGO, some wildlife professional. How do collaborations form? What are some of the successes and maybe challenges associated with forming those collaborations? And then probably the most exciting aspect of this is there's going to be an actual product to locate a geneticist for collaboration that is being developed as we speak. And if folks have more questions about this, Caitlin Atkan, one of our panelists, is the brainchild behind it in developing this. See if this should be loading. It might just take a second. I really hope it loads. I'd like to show you the format, but it's a way to try to, if you're in a particular region with a particular question, who can you reach out to, right? So this is in development, but we're populating this with molecular or TWS members and others to help bring those collaborations together and to help form, partnerships that incorporate molecular ecology when needed to enhance a project or a research question. So yeah, so please reach out to Caitlin if you have questions about that. All right, so lastly, before we jump into the Choose Your Own Adventure, I just want to highlight some of the really exciting work that some of our members do. Starting with probably one of the most emblematic wildlife management species in North America, right? The white-tailed deer. So there's a number of interesting projects being developed, particularly this one, with first one here in the bullet about a multi-state, 17 different states coming together to develop genomic tools to better manage CWD in white-tailed deer, which surprisingly, there aren't that many genetic or genomic tools available for this, really critically important management species. So that's going to have cascading effects. some other great projects looking at not using non-invasive fecal sampling to do molecular diet analyses, so kind of a cutting edge tool that's being developed for wildlife to be able to assess diet potentially more quickly or comprehensively than some other traditional techniques. And then, combining genetic marker capture. This is kind of the heart of what a lot of wildlife biologists do with traditional methods of radiotelemetry and in human dimensions, like these multifaceted aspects of wildlife biology to better understand this common species along urban and suburban gradients. But, we work around the globe as molecular ecologists. So international research, or at least international species, like in the case of African wild dog, where, or African painted dog, where WOG member is partnering with Pittsburgh Zoo and PPG Aquarium to do genetic assessment and genetic variation of captive wild dogs to inform the actual captive breeding genetic management, which has cascading effects for wild populations. Here in the middle, a PhD student from Dr. Lizette Waite's labs, who's doing the first genetic assessment ever of bats on the Galapagos Islands, right? Which might be a distinct species. There's nothing known about their ecology. So here's genetics informing the first step of trying to understand what's even there. Pretty interesting stuff. And then on the far right, here's a mandrill from Central Africa, where WOG members are using environmental DNA to understand broad community assemblages to understand this, threatened species and presence absence of it across the landscape. And so I guess the kind of overarching umbrella here is that we're a collaborative group, and we do interdisciplinary work just with the goal to enhance wildlife work, whether it's research, it's management, it's conservation. And I feel like through my time in TWS and other societies that sometimes people feel maybe a little intimidated to get into genetics, or to think that they could incorporate or understand genetics into kind of their tool belt of wildlife management. But in reality, wildlife geneticists are first and foremost wildlife biologists. And so we speak the same language and we want the same objectives. And so, at the end of the day, we want to look at cool animals and understand them, which is another tool to do that. And I think, that this group has been really great at creating these networks and being this resource to do these things. All right, so that was our quick, hopefully speedy introduction to WOG and what we are as a group. But what we really want to do now is spend the rest of our time together today going into topics that you as the audience are most interested in hearing about and learning about. So we've got an excellent group of panelists that are going to be weighing in on each one of these. And as you as audience, please use the Q&A when we get to that point to ask questions that we'll field. And then in just a minute, not yet, but in just a minute, I'm going to ask Akia to launch a poll where you can select your top two choices for us to go into and discuss. And the topics that we're presenting today, again, these are based on conversations and interests that we hear quite a bit about. First are logistics. So this is almost like a little condensed version of our Do the Genetics 101, but kind of getting into sample collection. What can genetics do and not do? And some of this general costs and considerations for starting a genetic project. The second topic you could choose is eDNA, or environmental DNA. In this topic will go into some of the really interesting cutting edge applications of getting DNA from the environment to study biodiversity or detect rare species or invasive species management. And again, think about some of the considerations about implementing a project like that. Disease management, so really integral to disease management is the genetic component. And so this section would talk about the genetic aspects of disease transmission and association, and kind of hammer home ideas about what samples are needed and what, again, can you do and not do with the genetic data. Next topic, potential choice would be omics, which oftentimes we think of genomics, so scaling up from a couple genes to an entire genome. And when is this necessary for management? And what new questions can you ask with it? Population dynamics is the fifth choice. So, you know, estimating size, relatedness, patterns of connectivity, these important parameters that are often a key point of understanding the dynamics of a particular wildlife population. And then finally, the last topic is beyond species and population management. So this is kind of a big topic here, but the idea is thinking about things beyond the individual and questions about hybridization and systematics and how did something become the way it is, the evolutionary questions that we also do as molecular ecologists, including things like historic demography in a population that could impact contemporary population fitness. So these are our topics. Akia, you can go ahead and launch that poll. I'd ask everyone to please select your top 2, and we will then choose our first adventure based on the top ranked topics.
Okay, the poll is live. Currently got about 90-ish, 90 people have voted. Let's see, coming up on the home stretch, so we've got about 106 or so people.
All right, do a countdown. 5, 4, 3, 2, 1, vote.
All right, I'll go ahead and end the poll then. I'll go ahead and share that with you guys. Can you see the polling results there?
We can. Thank you so much. All right. So make sure I can interpret this. Do all the panelists see the poll? And thank you, audience, for bearing with us with this. My first time using a poll.
Yes, Kristen, we can all see the poll.
Excellent. Thank you, Emily. So our first topic that we're going to jump into looks to be, what is that? Population dynamics. So I will stop sharing my screen here. And Liz, why don't you go ahead and share the population dynamics intro. So the general format that we're going to use as we go into our panel discussion is that one panelist has some introductory slides that they will walk us through that should help generate and guide discussion a bit. And then if you any questions you have, please put into the Q&A. I'll be monitoring that and getting those to the panelists. And yeah, way to go. This is pretty informal. Let's have a discussion and talk about things that are gonna help people in their next steps as molecular ecologists.
Hey, everybody can see it? Awesome.
We've got your, we're seeing your note slide version, if that matters.
Oh, I'm gonna, there we go.
There we go.
I don't have any notes on it. Well, so thanks for everybody coming today. And I have some experience doing some population dynamic stuff, mark recapture, landscape genetics, things of that nature. So just to give you, sorry, my dog wants to also join the presentation. And so there are lots of applications within population dynamics. Again, mark recapture, I think, is maybe the most popular method within population dynamics. You can use that to estimate population sizes, look at sex ratios, things of that nature. Go to your bed. And then also you can look at things like relatedness, estimate how genetically similar individuals or populations are. Look at spatial genetic structure, things like spatial autocorrelations and things of that nature, or barrier detection. So looking at, is a road a barrier to movement? And then also just as a field, landscape genetics. So looking at the landscape effects of connectivity and effective dispersal. Essentially, we're measuring effective dispersal, which means where the individual was born, where they ended up, and bred. So you can detect that through genetics. That's a little bit different when you're looking at things like telemetry, where you're looking at daily movements. And so this field often includes opportunistic or non-invasive genetic sampling. And so because of that, there are some considerations when you're dealing with these types of samples, because for these types of questions, you need a lot of samples. And so what that really means is that you have to either combine data sets. So for example, I've worked with a lot of carnivores where we'll use fecal sampling, hair sampling, roadkills, fur trap samples, lots of different ways to collect samples of species depending on which one you're looking at. You can also run transects or certain grids to sample fecal or hair samples. And if you're in different species from birds, like birds, you can use feathers, you can use scent marking. There are lots of different materials you can use, but I found in wildlife management, hair and fecal samples tend to be the most common. But there's also, because you need a lot of samples, this often involves a lot of collaboration with not just academics, but as well as your local hunters, your trappers, fishermen, sportsmen, just plain homeowners that may have an animal that they have trapped as a pest. I've worked with exterminators, basically anybody that will get you a sample and can get to you relatively fresh is a great way to get samples. And so with these types of really large data sets, you're going to have a lot of upfront laboratory costs, especially when it comes to optimizing. So because non-invasive samples are often low quality, that you have to acknowledge in your budgets that not all those samples are going to successfully amplify. Just unfortunately, there are a lot of different factors that go into whether these samples will work. A big one would be climate. So I'm in North Carolina. The success rates of my fecal samples are going to be very different than a place like Alaska, because very different climatic factors. You need to optimize these samples because, again, you want everything sort of set up front. So when you're processing sometimes thousands of samples, it's really important that all of that is done up front, so you don't have to repeat those samples over and over and over and over just to control for these. And then repeated amplifications are needed to quantify errors. In this picture, you can see something that's called allelic dropout. It's the bane of most people's existence when it comes to microsatellites. Excuse me. And essentially what it causes is you have an individual that has 2 peaks and above, and then you run that individual again, and now they have 1 peak. So they would look like different individuals, but that's just due to an error. And then finally, a big consideration when you're doing low copy samples is that sometimes they may not be quality enough to do some genomic questions. So you sort of have to consider what questions you can answer with the type of data you have. And so with all of those considerations, there are lots of different examples that use these different types of techniques. I'm shameless plug for me of some work that I did with mountain gorillas in Uganda using fecal samples. And so what this is used for is monitoring population sizes, group sizes, movements, or what they call transfers within gorillas within these parks, because gorillas, mountain gorillas are found in basically 2 parks and nowhere else. They're highly dependent on management to survive. And so what they do every 2 to 5 years is they walk these parks and literally walk the entire park collecting samples. And then they're run at either now SNPs, but at this time it was microsatellites. And we can estimate things like their population size changes over time, which it shows that they are increasing, which is great. Things like relatedness, troop size or group size, and as well as these different behavioral aspects of their biology, looking at do these aggressive encounters, do these females leave when a male dies and things like that, because half the population is very well known. So that's really all I have, but if there's any questions beyond that, I'm more than happy to answer them. I'm sure my fellow panelists would be as well.
Excellent. Thank you so much, Liz. That was a great introduction. And like I said, please feel free, anyone, throw questions in the Q&A. Oh, we got one in the chat. That works too. It will help if we can get it into the Q&A, just because we can track it a little better. So folks could do that. But first question, is there an example where a small sample size can be used in this area? So for population dynamic studies, can you use really small sample sizes and still get information?
I mean, I think the answer, it depends on the species. So sometimes you don't have a choice. So if you're using a small sample size on something like a white-tailed deer, it's probably not going to give you the answer that you really want. But if you're working with something like gorillas, for example, we had, we collected thousands of samples, but we still only got 400 individuals. In some cases, for barrier detection, you don't need that many. Again, it really depends on your question, but I would say sample size is completely dependent on that species biology and the question you're asking. So there are examples in endangered species, for example, like rhinos and things like that. They don't have a lot of individuals. If you're looking at relatedness, you may not need as high as a mark recapture study, because the accuracy of those methods completely depends on how many individuals you sample. So, and recapture. So if you have a species that is pretty rare and doesn't have a large population size, then you're fine with a small number of samples. If you're dealing with something like a white-tailed deer or a feral hog or something that has millions of samples and you want to sample them over a really large geographic area, then that might affect your analyses. But again, it really depends on how big your area is as well. If you're sampling within a fairly small geographic area, you're not going to need the thousands of samples that you would need for a statewide or a nationwide an assessment, if that makes sense.
And I can follow up on that too. I think if what you're trying to estimate is a population parameter, then you'll want to have a good sample of that population to get a good estimate of whatever that parameter is. And so if you're trying to estimate a population parameter, you need to have a good enough sample that you've well estimated that parameter. So if your population is only 10, then maybe you only need a handful of samples, right? You can only get 10. So maybe you get a pretty good estimate with 5 samples. But if your population size is 5,000, then you probably need a larger number of samples in order to be able to accurately estimate that parameter.
And I can add to that as well. So as Emily said, it depends on the question that you're asking. If you're looking at relatedness among individuals, then a small sample size of 5 or 10 individuals is probably going to give you the answer that you want, with a caveat that I'll come to in a second. But as she said, if you're looking at a population level question, population size, effective population size, that sort of thing, then you're going to need a larger number. My kind of ballpark figure that I always try and aim for is a minimum of 15 individuals per sampling location. And it does depend on also the markers that you're using and the level of variability in the marker that you're using. So if, for example, you're using microsatellites, there's a lot of variation in terms of how many different alleles one microsatellite locus has compared to another. And so that's where a discussion with the molecular ecologist that you're hopefully working with would be really valuable, and what background work has been done on that species. If there are lots of highly variable markers available, then you could probably get away with fewer individual samples, because you're still going to get the resolution that you need even with a small sample size. But if there are few markers available, or the markers don't have a lot of variability in them, then you're going to need a larger number of individuals to give you that resolution. And then one other thing I wanted to add to what Liz mentioned is in terms of the things to be aware of when you're collecting samples. Again, depending on the question, but it's not just the number of samples, but also where those samples came from. So if you're looking at population level questions, like how does this population compare to this population, you really need to know exactly where those samples came from. So if you're working with multiple people, with hunters, with trappers, with fishermen, etc., as Liz mentioned, you really need to be sure that they're going to give you accurate locational information when you collect those samples, so that when you do those analyses, you can assign specific individuals to specific regions and get good information from that.
I will add one final statement, I think, on this topic, and that is that it's important to remember you cannot just throw out all the rules commonly associated with traditional population parameter estimation just because you're using genetics. So, the number of samples you're going to need if you're doing like abundance or spatial capture, recapture, or something like that, those same rules apply if you were, actually out capturing animals. The only difference is now with, for example, if you're using non-invasive genetic sampling, some of those samples are likely to fail or will fail, right? And so you're going to need more than the number that you would traditionally need if you were doing live capture. type things.
So we have a lot of really great questions in the chat here. And I think one that maybe follows up on that, because Cassandra brought up what marker are you using? How are you genotyping? And someone asked, in the age of omics, what are your thoughts on microsats versus genomics for analysis of population dynamics, which I know is something that we all think and talk a lot about. So that's a great, I think, a relevant, timely question. Thoughts?
Emily, you've had quite a lot of experience dealing with both. Maybe you'd like to start off. Don't mean to put you on the spot, but
That's fine. That's what we're all here for, right? So, and I know, there's a lot of panelists and a lot of people in the participants as well who've got quite a bit of experience in this area too. And so I highly encourage them to jump in with their insight as well. But yeah, I mean, I think as the field embraces genomics, sometimes there's a tendency to go for the new and exciting thing, right? And so I understand that, and I am one of them, right, who often sees this as an exciting new tool. But there are certainly times where you might choose one option over another. I think sort of right off the bat, to sort of start the discussion, one thing you would consider is what is already known about your species of interest in terms of its genetics. That would be sort of my first question, is what species are you working on? And so for a species where we know a lot about its genetics and we know a lot about its genome, we might be able to use genomics more to better effect, right? We might be able to know a lot about how that genome is organized, a lot about what species it's related to, and have an actual reference genome sequence that we can use to develop markers and develop markers that can help us answer the questions that we really want to answer. If there's next to nothing known about your species, it's really hard to use genomics because we really don't know what to expect. That's not to say it can't be done. It just means that there's going to be a lot more sort of exploratory work on the front end. I also think that, the type of question that you're going to answer is really important too. If you're going to answer questions about like mark recapture, microsatellites work really well for individual identification. And so if there's a panel of microsatellites that's already developed for your species and you're going to do a spatial mark recapture and you're going to use non-invasive samples, I would go with microsatellites because they are really effective and they work really well. And you can do them on one or a handful of species time periods. So sort of the third component is that I would bring up is that it also depends on whether your study is sort of a one and done or whether your study is ongoing. So that would be the third major consideration, I would say, is that if you're going to be running one or a handful of samples often, I would probably choose microsatellites over omics, because with genomics, you very often, not always, but you very often sort of resurvey the genome new each time. And as you do that, you sort of lose markers in common across all of your samplings. And so you can really whittle down the number of informative markers that you end up being able to use if you've run your samples over multiple years or multiple sampling occasions. Whereas with microsatellites, you can retain the same number that you started with more easily. So those are sort of the three main things I would think of. And there's, a million more things that everybody here, I'm sure, has to add. So I'll let them join in too.
Thanks, Emily. I would just add to that when we think of it, it's exactly what Emily says, can be also sample type dependent, where we can do things easier with a high quality tissue sample than we can with a fecal sample, because you get different quality DNA from those things. And there's, I think, a lot of effort being put in right now into like SNP assays for individual species or populations that probably would be similar in utility to microsatellites, just a different process of amplifying and sequencing and genotyping. And so I would guess in the next handful of years, we're already seeing a lot, and we're planning to see a lot more of something like, whether it's a GT-seq, that's like kind of a hot thing right now, or Fluidigm, or there's other techniques out there that people are actively developing to kind of go hand in hand with with how microsatellites have been used, you know, the last 20 years. But neither ever replaces other techniques. It's just more things, more abilities and more tools and more applications that we can do as new techniques come online. All right. Let's maybe do one more question. I will keep these Q&As up because maybe afterwards panelists could even go and see if they want to type some answers in as we move on. But if we don't move on, we're not going to get to a second topic. And I don't even think we're going to get to a third topic. All right, because you guys are asking excellent questions. This is great. All right, so I'm going to go with the and in order here. So could you give a quick overview of how effective dispersal would be estimated with genomic samples? So kind of a you know, population dynamic question of animal movement and dispersal. Anyone work with that? And maybe before we dive into that, I'd just like to mention some of our other active molecular ecology working group folks are making some really good discussion points in the chat that all of these considerations always also have a price tag. And so at the end of the day, we may want to sequence a whole genome, but we can't do that on 100 animals. And so instead we choose, so right, part of the consideration is going to be always the actual ability of the money and the finances and the resources you have to do something, right?
So I guess I can do the effective dispersal one. I've done a little bit with Gray Fox because it's dealing with a hybrid zone. So I'll preface this that these two groups are highly differentiated from each other. So it's much easier to do when you're looking at effective dispersal. If you have separate populations, you can use a whole host of different techniques versus if you're looking at within a population. Because if you have two differentiated populations like I had in the study using omics, I can track, well, this individual very much belongs to this one area that this other population has found, and I found it in this new area. So it obviously, that was its effective dispersal. There's certainly techniques that you can indirectly estimate effective dispersal. If you're going to do like true effective dispersal, you probably need where mom is and what area she's using, and then you need where her, without a doubt, offspring was found. So you can do that through parentage and things like that, which can be used as either microsatellites or omics. Omics gives you more power usually in those cases. But there's a host of methods that can do these between population techniques. So things like the program structure or fast structure in this case, or things like that, where you can take an individual, where these are Bayesian clustering programs. So you have these different sets of populations that you hypothesize that are there. You determine the number of populations, and then when an individual is trapped in a different area than the rest of their cluster or their population, you can say, well, that effective dispersal, they've clearly moved to that new area. Within populations, it gets more difficult. As I mentioned before, I think parentage is probably the best way, but it's also probably the most difficult. I'm sure people who are much more experienced with parentage can attest to that, just because it can be hard to, without like high probability, especially in large populations, to assign parent offspring pairs and differentiate them between siblings or something like that. So I tried that in bears, for example, and we had a little trouble because bears live a long time. And we had one that was older, but we couldn't determine, was that bear that was 5 years old the son of, or was he a younger sibling? So that's where it can get a little bit hairy. But if you do have, a lot about your population, that will definitely help with estimating effective dispersal without an indirect method. An indirect method, there's things like spatial autocorrelation and things like that that can give you the scale of your dispersal across the population, but individuals gets a little bit more difficult.
Can I just say one thing that's a little off topic? It's more on the omics versus microsat conversation, but we talked about quality of sample and how much funding you have. Those are considerations, but also the consideration is like your bioinformatic ability and also your computational skills. So like with omics, a lot of times you kind of require like a higher processing computer or a computer cluster to kind of do these large genomic analyses, and also the ability to do that, using the Unix command line. So there is a little bit of a learning curve there to actually go over that that hurdle to kind of go into the bioinformatic bit.
Yeah, that's a really great point. And in the chat, some of our members are suggesting also how that transition from microsats to omics is really pushing the envelope, right? So this is being done in human genetics and biomedical fields and stuff like that. But on the ground wildlife management is very different, right? And it takes a while for these things to trickle down. One, because they might not be necessary. And so whether or not omics is going to become the bread and butter, this is from Tony. She's saying, maybe, but maybe not. There's always something new. It takes time to vet these techniques. It takes time to do the comparative studies to see what is going to be most effective and filter down into our on the ground world, which I think is a really a good point. This is great. So let's move on to our second topic. And I was just going to follow up. Rachel made such an excellent point about bioinformatics and clusters, and that's another beauty is like probably a lot of people on this call have those or have access to them. So then you get to reach out and get the help with that aspect of a project. If you can generate the data, but you need a collaborator to help run it, right? So again, that's kind of what we're here for. All right. And we cleared all our questions. Excellent. So the next topic, if I copied this down correctly, was eDNA, which I will share my screen and get started on that. But please recognize this panel is designed that none of us are the experts, right? We're all here together to discuss, and so join in, panelists. Okay. So eDNA was the next selected topic. So eDNA, environmental DNA, the definition is, right, it's a mixture of genomic DNA from many different organisms that you get from the environment. And this could be any sort of environmental sample, air, water, invertebrate, species, soil, it's kind of an endless list, leaves, that you can get that environmental DNA. But the distinction is it's not from an individual, right? It's not, I'm going to go sample this individual scat, this species scat or tissue or something like that. It's a community from the environment. And this is a really growing field, and the applications are becoming, you see more and more every month with this, and to the point that there even has its own journal. There's an environmental DNA journal dedicated to the study and use of environmental DNA for basic and applied science. And there's a ton of different eDNA research out there. I kind of feel like it kind of gets distilled down potentially into two main flavors, where you're trying to detect the presence of a single species from some sort of environmental sample. So this could be a rare or invasive species, or you're trying to detect the presence of an entire community or look at the biodiversity of a system. So the single species detection or presence of all the species. And to kind of walk you through the basic workflow of what this is you go and you collect some sort of environmental sample. In this example, it's a fly. You do a DNA extraction, you sequence or do some sort of amplification of that extraction, and you learn about the community that maybe that fly, that carrion fly, was eating, dead carcasses of all the vertebrates in your community. You can do this to, advancing to detect that single species versus the whole community. It can be from a variety of different environmental sources. Some of the very common techniques right now include DNA metabarcoding, which is a very small genetic sequence that can define things down to specific taxa. Or you can use species-specific markers to identify like an endangered species or an invasive species. This work has been done a long time in the aquatic system, and in the last I'd say 5-ish years, is becoming more prevalent in terrestrial systems. So just to give a couple examples of within these two flavors of some really cool research, one studying with presence of single species, detecting rare or invasives. There's a lot of great work done. I like this paper by Williams et al. 2017 demonstrating that you can detect invasive feral swine from DNA that they leave in wallows, right? So by going and sampling that water, you're able to understand if there were feral swine there. This is common applications in aquatic systems. You see this a lot in fisheries. Really exciting paper that came out a couple years ago was detecting rare carnivores from snow tracks. So go collecting their actual tracks and keeping that snow and extracting DNA from it, being able to identify that individual species. Then example of scaling up to looking at the presence of all species or community biodiversity. Some work you see a lot in really biodiverse regions, so a lot of tropical regions, are collecting some sort of invertebrate. Like here's an example of a fly trap that you collect carrion flies in. You can go and set up a bunch of camera traps, which we're all pretty familiar with, and then compare the communities that you get on these two methods, because this really comes back to a lot of those considerations that we talked about of, time input to go put up 50 cameras in a really remote area and have to go service them and get the batteries, versus maybe setting up a fly trap that you leave for 24 hours and or even 10 minutes or something like that, and it's much faster potentially. And so there's a Rogers et al. Paper comparing in a very well studied population in Barrow, Colorado, Panama, looking at combinations of carrion fly, eDNA, to camera traps and other transect counts. And the take-home message is that carrion fly is good, detected more species in small bouts, but then long-term camera deployments actually detected more. And so it's really a combination of these techniques. So again, just adding more tools to your tool belt to study a population. So some of the considerations, a big one when we think about lab work is contamination, because we're detecting such tiny amounts of DNA, and we're using genetic markers that are universal in the sense that they amplify all taxa, or all vertebrates or all mammals, that our DNA would also be easily amplified. So contamination is a really big deal. And as such, if you want to do this type of study, unlike the potential of getting roadkill, you can't just kind of haphazardly say, I'm going to go sample something for eDNA. It needs to be pretty specific, objective, and designed to prevent contamination and make sure you're storing samples properly. I think I listed a kind of a variety of considerations here, but really like a big one would be comparison between methods. I think this is a really active area of research right now, as these different methods apply to our work as wildlife biologists, whether you're measuring a community or trying to identify that one individual. Is that molecular method better, worse, or equal to maybe or complementary to other methods that are commonly used? So with that quick introduction, I'd love to open up a conversation about eDNA, because like I said, I think it's a really exciting tool that is going to increase in application. I see in the, I don't know if folks are monitoring the chat, but Rob mentioned how that really, well, Rob, why don't you just say, I don't need to read your chat.
No worries. Yeah, I was just saying that another really contemporary example of eDNA, not for wildlife, but monitoring humans, is actually using sewage and testing sewage to monitor the sort of change in infection rate in the community for COVID. So that's been used on many campuses and in some cities, and effectively could be that same concept, right, can be applied to wildlife populations looking at disease as well.
Great. Well, we're having questions roll in here. I can read off the first one. If anyone online has done anything with owl pellets, can you evaluate the genetics of an individual owl using an owl pellet? Or would owl pellets be better suited for like an eDNA approach to detect prey species? I see in our chat that Tony, can you unmute yourself? She doesn't know. Well, I'll read her chat because she's making really good points because she's done it with vultures. So she's saying that they've done it with vulture pellets and doing it both ways. So tracking the individual vulture and then also the diet of the vulture. So well, I haven't done it with owls, given you can do it with vultures. I can't imagine why you wouldn't be able to do that, given that the metabarcoding for the diet. Okay, Tony's in.
I'm here. Thank you guys for giving me that. I was like, there's no button. There's only a couple owl pellet papers, and typically they have only been used to look at diet. So I think that's a really great question, because I think it's something that would have a lot of utility, which is why we did the vulture pellets and developed vulture microsatellites, again, our bread and butter, to track vulture populations and then also look at the diet. Thank you.
Just a comment on the chat real quick. We can change the chat function so it's not just to all panelists, that's the default. So you can do all panelists and attendees so the attendees can also see our chat. Just throwing it out there.
Got it. I hadn't seen that. Thank you.
Great. All right, so some other questions that have come up here. How big is the issue of false positives and metabarcoding, particularly when the species you're interested in might be rare? Tony, it sounds like you had some good experience with the diet work. Would you get, I guess first, I'd love to hear other people's comments, but say, one, it's hard to detect that because you don't know, right? You don't know what's in your sample. So there are strategies and methods for having a known community, like positive control, that you include in your genetic prep. And that is one way to try to track things like false positives or unequal amplification between taxonomic groups that might lead to, the wrong kind of community composition. But it's tricky, and it's a consideration.
Absolutely.
Can I add to that? So go ahead, Tony.
No, I was going to say there's really clear studies that show there is always a level of contamination within metabarcoding studies. There's a really good air study, and the guy's looking for really low quality DNA, and it's just really often very hard to parse apart the signal between what's real and what's contamination, because you're going to pick up things that are in your lab and floating around in air. People have put swabs out in a lab. But that's not to say, again, it's the question that you're asking. So if you're looking for the common things in a diet or the common things in the water, instead of the rare things, then it's a really good method even right now. And a lot of people are pushing the envelope again on trying to figure out how to get the rare things. One thing we're doing with feral swine diet is using CRISPR-Cas9 to cut out the feral swine DNA so we can get at the other things that are in that fecal sample, because you can imagine how many copies of that feral swine DNA you're going to get, which can swamp out those rare things, which is often what you're looking for, honestly, right? The endangered species that pig is eating. So that's all.
And I can speak to a little bit about the prevention of, or at least, so contamination's really hard to prevent completely, but the importance of running controls within these to identify, so that's a sample that shouldn't have anything in it. And if you're getting positives from that, that's a really good way to identify contamination. There are code and things like that to remove things that are extraneous. I'm working with some people who are doing microbiome stuff right now, and it's a pretty similar, they deal with bacterial contamination, which is a whole different animal, but, or not animal. And so there are definitely different ways to assess contamination, both computationally as well as in the lab. But unfortunately, just because if you're doing with fecal samples, for example, it bounces in the dirt. There can be things in the dirt. You can't prevent that. So really, it's just to determine. Those are a couple of ways to help determine. But yeah, you're always going to have it. It's just, you know, there are many.
I know we're here talking about genetics, but I'll just put in a quick quantitative sort of note as well, right? So we've talked about the fact that, you know, we can deal with it some degree by having controls in the lab, and Liz just commented that there's some quantitative techniques, right? So often with eDNA, we're looking at species level patterns and species level occurrence, not individuals. And so if you're collecting this data with the intention of using it in something like an occupancy model, then it might be wise to have some alternative sampling design as well, some alternative sampling technique where you have some sites of known occurrence so that you can explicitly model false positives within an occupancy modeling framework or something like that. So there's ways to handle it on the front end, but also some ways to try to account for that on the back end as well.
And as Rob rightfully points out, a lot of those come from basic quantitative ecology and wildlife management that everybody knows, whether you're a geneticist or a non-geneticist. They're these same techniques and they're these same ideas, just in a genetics context.
All right. I mean, we are like 3 minutes to the end of our webinar. This went incredibly fast. And there's still a lot of really great questions. So I think that Molecular Ecology Working Group should just do a webinar for our working group in like a couple months. So we just do this with a couple topics, and we'll make sure we advertise broadly to the TWS, because I think this went really well. So one final question. I can't pick. There's many good ones. I feel like we touched on a lot of these. I can't pick. They're all good. Yeah.
I just want to let you know that we can go over time to clear the questions. There's no restrictions with that if you wanted just to clear all of the questions.
So I will not require everyone to stay on, but if folks want to, I'm happy to answer a few more questions and then clear questions that we have. I'm happy to do that. All right, so where did we leave off then? We talked about false positives. We talked about some contamination issues. I'll just say my lab group now always wears masks when they do lab work exclusively, even outside of COVID restrictions, just for prevention, particularly with eDNA of sample contamination. Can you use a CO1 gene to create an eDNA marker, or do you need to obtain a live sample? For example, Eastern Masaga CO1 gene has been mapped out on GenBank. Could we use this instead of getting a sample? So can you use a reference sample to design some sort of metabarcode primer pair? Yes.
Kristen, can I just hop in? Sorry, it's off topic from this question, but I was also just thinking about the contamination question coming back. So, we've mentioned, and you mentioned working in the lab to prevent contamination and running negative controls like Liz mentioned. And, I routinely work on a hood to do my PCRs and so on. But a major step of preventing contamination is at the sample collection stage itself. So if you're out collecting, especially a really low quality sample, like a fecal sample or something, it's really essential that obviously you wear gloves. If you're using, say you're scraping the outside of a scat to collect, sorry, my dog is now wanting to join in. Make sure you use a different scalpel for each sample. If it's a tissue sample, make sure you're using a different scalpel, or at least you sterilize with bleach or something from one sample to the next to get rid of any DNA. So you have to be aware of contamination issues right from the get-go. And especially, I haven't done much eDNA work myself, but human contamination of your sample as well is important, especially if you're using markers that could cross-amplify in humans. So that's something to be aware of, your own DNA contaminating the sample, and then one sample, if you're doing bat samples, wing punching, you're using a little thing to punch out a sample of tissue, make sure you use a new one each time, or you sterilize it with bleach to get rid of the DNA from sample 1 before you use it on sample 2. Sorry, you can go back to the CO1 question now.
Well, that was super important, because we all know how messy field work can get, but with eDNA, it really needs to be a priority that gloves are being worn and you're using best practices for an attempt of sterility and cleanliness in the field. Yeah, so CO1 gene to create some sort of metabarcode from a reference sample. So yeah, you can use a reference sample to design a primer pair. You'd probably, anyone else, please jump in too, because I haven't done this for a metabarcode, but it's the same. There's a lot of considerations, and in fact, if you're really interested in doing this, I would suggest finding this book or emailing me. And there's a whole section on designing metabarcodes and the considerations that you want to think about, because they're a little bit different than your standard primer designs when just designing a standard primer. And a lot of there's a whole chapter and about designing your metabarcode. But if you have a reference sequence, you can design a metabarcode. All right. Are there any recommendations for detecting very low prevalence density of potential environmental contaminants, i.e. oocytes, without sampling huge quantities, for example, water? And I can't answer that. Has anyone been doing disease detection? I feel like Tony was, I think she left us, but Tony probably could have answered that, I think.
The only one that I've done is ranavirus, and it was too common probably to assess that. So it was pretty common and they were vernal ponds. But I mean, I can ask, so we do have a fish eDNA person that I collaborate with here at the museum. I can go and ask them if you want to give me your contact information, but I'm not sure. I've got a thunderstorm right now, so.
With detecting plague, which usually it's done using fleas, which is pretty low quantity for the bacteria. Usually it's like a pooled approach. So you like pool a bunch of the fleas from a single area. So you kind of like have more DNA to work with from multiple individuals. And then you can do like qPCR for detection.
Which I don't know if the CO1 person's still on, but if you're doing single species detection, you're using qPCR. And so there's other considerations for that as well. All right, last question. Does anyone have experience using eDNA to survey for current occupation of small freshwater fish in shallow watershed tributaries? I do not. But I know this is a often applied technique for which there's lots and lots of literature, that if someone was going to approach me, I would go find some of those people, but then I'd also look at that literature and there'd probably be some somewhat standard methodologies that have been worked out for this type of question. I think finding someone who's done it then could give you all the special little tips and expert tidbits that you only learn by doing something like a bunch of times and doing it wrong a bunch of times, and then you know how to do it right.
Yeah, and I'll just add that, like I'm not sure exactly what the interest is in it, but there's a lot of work that's been done on looking at like how long DNA persists in the stream, how water flow influences detection, distance to detection, how density of the fish or amphibian or whatever it is you're looking for, how density and activity. So when I say activity, like whether they're breeding or not breeding, how those all influence your ability to detect it. So there's a lot of different components that go into that. guess that's all I have to add.
So we do have a fisheries person in the chat right now that's answering. I don't know if Peter, if you can unmute yourself and talk, but that might be handy. I can read the, oh, it looks like it went out to all attendees as well, but in the chat there's an answer.
Hello, I guess I can unmute, or somebody unmuted me for me. I'm here. Yeah, I did a bit of eDNA metabarcoding work. And I mean, broadly, I think it just mirrors everything that Rob was saying, that there's a lot of different ins and outs to it, but most of the sort of basic approach is you go out and take multiple replicates of water samples in streams, doing it over time since the water is obviously moving. So you're going to have different sort of DNA there at different times. And then usually using 16S and 12S barcodes work pretty well for a lot of fish because they have a little bit more diversity sometimes than, or a little bit less diversity, I think it is, than CO1 to try to identify the species community that way.
I think we cleared our questions. All right.
Okay.
We're done.
All right, I'll go ahead and wrap up here. Thank you all for coming to our second webinar in the Wildlife Society webinar series. This recording will be posted to our website along with the Q&A questions. So please keep an eye out on the website for that recording. Also, please keep an eye out for updates about the July webinar that will also be going up on the website. Again, thank you all to the panelists, the Molecular Ecology Working Group for putting this all together. Thank you all for joining us as well. Yeah, have a great evening and a great rest of your day.
Bye, everyone.
Bye. Thank you.